and they make up to 30% of all polypeptides in eukaryotes, so like, for example, in humans. These disorder proteins do not possess defined native structure, but they assemble many different conformations. And this enables those proteins to bind to multiple targets, and they can fold upon binding. It has been found that they are abundant in diseases, for example, in Alzheimer's and Parkinson. There we can find these intrinsically disordered proteins. Now, when it comes to dynamics, the dynamics of structured proteins is quite well known, and in the simplest case, we can describe it as consisting of a local internal motion and a global rotational motion. The concept of a global rotational motion cannot be applied to intrinsically disordered proteins, because what is it that does the global rotational motion if we have kind of a rope that wiggles around and changes its shape all the time? So the question remains, how can we describe the dynamics in intrinsically disordered proteins? And this was the goal of the current study. So we investigated the dynamics of the intrinsically disordered protein with 15N NMR spin relaxation and with molecular dynamics simulations with different water models. And we did this in a temperature-dependent way. We investigated the dynamics in the temperature range from 5 to 55 degrees Celsius. Now, the IDP that we were investigating is the 26 amino acid long tail of the histone H4. So what is a histone? A histone is a protein that is important to wrap up DNA inside a cell. So DNA in humans is a 2-meter long molecule, and it has to fit inside the cell nucleus, which is only 10 micron. 10 micrometer big. And what happens is that the DNA is wrapped around a small protein particle, so-called histone, and this is shown in this picture here. So you see this is the DNA, and this is wrapped around, and here is a core particle, which consists of 8 histones, and there's 2 histones H4, which are shown in blue and in green here. And like here is the tail of the histone H4, and this is the protein that we were investigating. This 26 amino acid long sequence consists of 2 parts. There is a glycine-rich part. All glycines are marked here in red, so you see there's a lot of glycines. And there is a charged part. And this charged part is thought to be important for the compactation of the DNA, so that it fits into the cell nucleus. And this is, we expressed this protein, we purified it, and then we formed liquid NMR. First of all, we did the assignment for the protein so that we can now we know from HSQC type spectra which peak belongs to which sequence. We then measured spin relaxation, and the 2 parameters that we determined were the longitudinal relaxation rate R1 on one hand, and the dependence of this R1 as a function of the correlation time is very well known, and it's shown here just to remind you how it looks like. So this is this well-known shape that has a maximum at a certain correlation time. And then we determined the transverse chemical shift anisotropy dipolar cross-correlation rate beta. And this has a dependence on the correlation time, which is similar to the the transverse relaxation rate R2. But the advantage is that there is no dependence on the exchange. And how on chemical exchange now. We determine this cross-correlation rate beta with a proton-coupled spectrum, HSQC type spectrum, in which we observe for each NH 2 peaks, which are separated by the NH-J coupling. Now, from the intensity ratio of these two peaks, which is shown here, we can determine the cross-correlation rate beta when we vary the evolution time delta or delta. So this is the experimental points. We can fit it and get the cross-relaxation time. Here are the NMR results at 25 degrees Celsius. In the left top picture, there's the longitudinal relaxation rate. So the points are the intensity as a function of the evolution time for 3 different residues. And most of the residues do not scatter as much as like this first observable peak. Most of the residues give very smooth curves. So we can fit it and we get R1 values with small errors. And here you see examples for the same residues, the CSA level across correlation rate. In the lower left picture, you see the temperature dependence for the same residues, and we see that with higher temperatures, the relaxation rate goes down. This is the same for CSA, so we know that the dynamics accelerates with temperature, and this is what we expect, of course. In this last picture on this slide, we see the relaxation rate as a function of the residue. We can determine the relaxation rates almost for all residues in the sequence, and we can distinguish 2 parts. The glycine-rich part moves faster and has thus a smaller relaxation rate than the charged part. Now we perform molecular dynamic simulation of the same molecule in a box of water. And the simulations were performed with the software Amber and in the force field, which is relatively new, force field FF14SB. All simulations had a length of 2 microseconds, and from the simulations we can calculate the correlation function of backbone nitrogen-proton vector. From this correlation function, it's possible to calculate the NMR parameters. In order to do so, we have to assume a motional model. The simplest motional model that fits the correlation function is a 2-component motional model, which consists of a fast and local motion and a slower, larger motion. Here you see an example of the correlation function in brown and the fit in black. And from the fit, we have now these 2 components. So each component has a weight, which is on the y-axis in this picture, and has a correlation time. And you see here the temperature development of the weights of the fast and the slow component. And here we have the representation of the relaxation rate as a function of the residue, which again shows we have faster relaxation in the glycine-rich part and slower relaxation in the charged part. We performed the simulation with different water models and wanted to know which of the water models leads to the best reproduction of the experimental values. The water models that we used, the water models in principle have been shown to influence the compactness of the structure. We used 2 water models, SPCE and TIP3P, which are so-called 3-point water models, in which a water molecule is represented with 3 points, so the oxygen here and the 2 protons there. But they differ a little bit in the angle between the protons and in the length of the oxygen-proton bond, which is shown here. And we also simulated 2 so-called 4-point water models in which the charge of the oxygen is a little bit shifted from the barycenter of the oxygen molecule. The 2 models that we used were TIP4P eval and TIP4PD. The model TIP4PD is a new water model which has been proposed by Piana et al. In 2015, and it is specially developed to model intrinsically disordered proteins. The authors found that in order to represent the characteristics of IDPs in a better way, it is necessary to scale the Lennard-Jones interaction, to scale the parameter epsilon here, and to make it much bigger than in the other water models. We can see the effect of the different water models on the compact structure when we evaluate the radius of gyration. Here there is it is shown the distribution of the radius of gyration, which is sampled along the 2 microsecond trajectory. We see that for the standard water models, which are SPCE, TIP3P, and TIP4P eval, there is a preference for very small radius of gyrations. So we have like a 2-hump distribution, so and at least there's a preference for the compact structure. While for TIP4PD, this preference is relieved, and we have a symmetric distribution, which can be, which is expected for freely moving disordered proteins. And because we find the same trend in the mean, when we plot the mean of the radius of gyration, and we see that for TIP4PD there's a larger value of this mean. Now we can compare the calculation of, we can look at the trajectories just to give you an impression what the difference is. So I'll show you 2 videos. Here there's the first of standard simulation in a standard water model, and you see that very often a compact structure is formed, and then sometimes the molecule extends, but it comes back and takes a more compact structure very often. We can compare this with the TIP4PD simulation, in which the molecule is more extended, but also forms sometimes small compact structures. But you see, yeah, it is in principle most of the time more extended than in the SPCE simulation. Now we can compare the simulated relaxation rates with the measured relaxation rates at 25 degrees Celsius, and this is done here. Here, this is the picture that you saw before. This is the NMR spin relaxation data as a function of the residue. So you have these 2 parts, and here we see the relaxation rates from ND simulations for all 4 different water models. And you see that the classical water models all produce relaxation rates that are too high, that are much higher than found experimentally. So the new water model TIP4PD reproduces the experimental data best. Now we can compare the temperature dependence in NMR and in MD simulations with this text model, and this is done in the first line for the relaxation rate, R1, and in the second line you see the cross-correlation rate, and you see that the temperature dependence is qualitatively met, and so we find that the MD simulations describe the experiment quite well, and we can use them to analyze the origin of motion in more detail. Yeah, I introduced the motion model that we used, and that we have two motion components. Yeah, we know that the origin of fast motion are vibration of dihedral angles, but it's not clear what is the origin of the slower motion. There's two different possibilities. In principle, it can be that the whole peptide motion, like a folding, unfolding type of motion, is responsible for the slower motion, or that it is only a motion of several amino acids that leads to this slower component. And now the question is, what is true here? And we can look at the simulation and just look for good examples. And this is shown here. What I showed you is the NH vector, which is the blue-white combination here, at two different points of time in one H4 molecule. And the 2 points in time are different by about 500 picoseconds. So this is the time scale in which the motion takes place. And you see that the NH vectors point in opposite directions. So the correlation between them is completely lost. But you can see in this bigger picture here that the overall conformation of the peptide is retained. So the correlation is lost, although the motion did not form any folding, unfolding transition. And now this is just one example, so it can be just accidentally that we see it here, and we wanted to know whether this is now this is true, whether we can find it in a statistical relevant way, and for that we performed some kind of Gedanken experiment, which I would like to explain on this slide. We wanted to know how many amino acids move together. So in principle, this is the question of what happens when I fix one amino acid, how long is the distance until the amino acid move freely again. So what we did is we fixed one amino acid. So here it is the number amino acid number 6, and we aligned in the simulations the atoms C alpha, C, O, and N. They were always put on top of each other. And then we calculated the NH vector correlation functions for this for the fixed molecule, which is of course not moving because it is fixed, so the correlation function does not decay very much, so we get a very high and constant value. And then we calculated the correlation function for the neighboring NH vectors. So here for amino acid 5, the correlation function decays a little bit, but still it is fixed because it is neighbored to the atoms that we hold fixed. And we did it to both sides, so for amino acid 5, for amino acid 4, for amino acid 7 and 8, and we can see that the larger the distance is to the fixed plane, the lower drops the correlation function. And in a distance of 2 amino acids, we almost find the same drop of correlation as in a non-fixed and free molecule, which is shown here in green. So that means that the distance, or no, the length of the peptide that is moving in order to lose the correlation, to lead to a correlation drop until 0 is only about 5 amino acids. This shows us that it is not the whole peptide that needs to move in order to explain the second component of our motion, but it is just the dihedral angle jumps in several peptide planes. Now, with this, I would like to sum up. I showed you today what happens, our investigation of the H4 tail protein. We performed NMR, relaxation measurements, and ND simulations. We compared the two techniques and found a agreement, even in the temperature dependence. And we used the molecular dynamic simulations to get more insight into the motional mechanism of the dynamics in an intrinsically disordered peptide. And we find that a two-component model fits the temperature-dependent well, and it is explained by fast, very local motion and a slower motion that involves several peptides. With this, I would like to acknowledge the financial support by the Russian Science Foundation and the support by the research part of St. Petersburg State University. And we used the resources from the Center for Molecular and Cell Technologies, we used the Computer Center, and we did all the NMR measurements in the Center for Magnetic Resonance. Yeah, now I thank you for your attention, and I'm open for questions.
So,
yes, I can hear you.
Kerstin, thank you very much, and we will try to ask you maybe. Please.
Hi, Kerstin, this is Nikolai speaking. I thought it was a great talk. One thing that crossed my mind, would we go back and look at the comparison of different water models, the relaxation rate profiles?
This one?
This one. So I'm looking here at the profile for tip 3P and the other one for tip 4PD. So the way that it looks like, the tip 3P actually reproduces the shape quite nicely, right? But everything is overestimated for the reason that tip 3P now produces this picture of a peptide which is too compact, too clustered, too viscous, right? This type of thing. So everything is qualitatively correct, but kind of systematically off. And then if we look at tip 4PD, it looks like it fixed the problem at the left shoulder, right? This glycine-rich portion. This is now utilized. It kind of loosened up. It's now very mobile. But the right portion actually remains too compact, right? And almost at the same level as tip 3P. So it seems like tip 4PD solved part of our problem, but didn't solve it entirely. Is this correct?
That's correct, yes. That is what I also think, yeah.
Thank you.
Sorry, I can't hear you.
We are fine.
Okay. Yeah, I think this is correct.
Any more questions?
Hello. Do you think that tip 4P water model will be the best for only intrinsically disordered protein, or maybe for another proteins too?
let me tell you a little bit more about TIP4PD. I don't think that TIP4PD is the best for intrinsically disordered proteins because it is just pointing in the right direction. So the authors just played around and tried to get a better representation and tried to find the origin of the misrepresentation of the standard water molecules. And they found that we have to tune the interaction that I showed you in order to get a better representation. So I think it is possible to develop even a better representation of water for the modulation of intrinsically disordered proteins. And but I think I don't know in which direction you're thinking. It is probably necessary to tune the water models for different applications. So it might be that we need some specialized water models for the perfect representation, let's say for DNA or something. Does it answer your question?
Yes, thank you.
Thank you. One more. Thank you very much, Kerstin, again, and I really wish you let's return. Let's return to